I did post the lecture recording for the last lecture in Canvas. So you should be able to see it. If you wanted to see it, it's there. It's just a direct recording. It's a screen capture of my screen. So if there was something you missed or you wanted to watch something again, those are always going to be there. Yeah, just make sure they work. But it should. All right. So we are going to oh, sorry. Would it help if I plugged it in? Okay, there we go. So we left off last time, we talked about ancient thinking and the ancient beliefs of people as it relates to microbiology and microscopic organisms. So today we are going to kind of jump into the discoveries that went into microbiology being the field that it is today. And again, just as a reminder, you never have to remember dates or numbers. I'm never going to ask you how many million bacteria are in a drop of water or anything like that. You don't have to remember that, but you do have to remember the discoveries and the people, okay? So hint, hint, wink, wink, there is going to be a question about the Cloaca Maxima. You don't have to know what year it was, but you have to know what it was, okay? And we're going to talk about Louis Pasteur on this slide. You have to know who Louis Pasteur was and what his contributions were, but you don't have to remember the date. I put the dates up there to give you some context to all of this, because I think it's important to realize that this wasn't all happening, thousands of years ago. 1859 wasn't that long ago, okay? Less than 200 years, which seems like a long time, but in perspective, it's really not that long. So now, the prevailing theory of current microbiology is something called biogenesis. But we're going to talk about what the theory was before biogenesis was proven. And biogenesis is the idea that life comes from other life. Again, if you look at the word, Genesis means creation of, bio means life. Like biology is the science of life. Biogenesis means that life is created from life, right? You look at the word. Now, that is the current prevailing theory, is that life comes from other life, right? And that should make sense to you. That should be pretty intuitive. Other forms of life are born or come from other forms of life, right? Humans don't just appear out of nowhere. They are conceived and incubated and born, right? Even bacteria are born from other bacteria. Things don't just pop out of nowhere. But for a long time before biogenesis was a theory, before that became the prevailing idea, there was the idea of something called spontaneous generation. And another word for spontaneous generation is abiogenesis. And remember, like we talked about last time, when you put that A in front of a word, it means without. So abiogenesis means life created not from life. Now, when we think about that now, that seems ridiculous, right? We know that things don't just pop into existence, right? Nothing does. Bugs, plants, they don't just pop into existence. But if you look at this from the framework of the 1600s, the 1500s, the 1400s, before that, they had no idea that there were things that were smaller than we could see that existed. So things like bugs showing up in grain stores, right? Weevils showing up in flour, that seems like something is popping out of nowhere. We know now that the bug larvae was already in the grain stores. They just hatched, but they appear like they're coming from nowhere. It's the same thing with like tadpoles in a pond, right? Or tadpoles in a puddle of rain. I have big, huge divots in the road in my subdivision, and every time it fills with water, there's tadpoles in them. Okay, it takes like 12 hours, and then there's tadpoles. If you didn't know that tadpoles are the reproductive unit of frogs, you might think that those tadpoles just appeared. They spontaneously generated out of nowhere. Because if you didn't know that there was something too small for you to see there, that's what it would look like. So spontaneous generation now seems like a ridiculous theory, but in the framework of what they knew back then, it's really not. So the prevailing theory through most of history was spontaneous generation or abiogenesis. Those two mean the same thing. And it's this idea that things, living things, can just appear. Okay, they didn't have to come from something else. They could just appear out of nowhere. Now, Louis Pasteur was the first scientist to definitively disprove abiogenesis. Okay. And he was the first really, really, truly important scientist in the field of microbiology. He was the beginning of this idea that there were tiny, tiny, tiny organisms that were spread from place to place. And the experiment that he developed to disprove spontaneous generation is a very famous one. It's a relatively simple experiment as far as experiments go, but it was very elegant in its simplicity. And to this day, it's probably one of the most important scientific experiments that was ever done. And so what he did, the way he did these experiments, is he created these special flasks. Okay, he hand blew these himself. He made them himself because nobody else was making them. And what they were just a round flask that he would put liquid in. And they had these big, huge curved necks. Okay, so you can see here, this curved neck flask. It was also called a swan-necked flask. And that big curve, what that allows to happen is it allows air into the flask, right? Because air is not a particle. Air can flow in there. Air can go through the curve and up and into the liquid part of the flask. But what can't go through the curve and back up and down is any kind of particle, whether we can see it or not. I know it's weird to think about, but bacteria have weight. They are a particle. They are a thing that has substance. They are a physical thing. They're very, very tiny, and they're very, very small, and they don't have a lot of mass, but they do have mass. They are a physical thing. And so anything that was in the air, any dust or bacteria or viruses or anything else would get caught in that neck, right? Because they would fall in the top, and they can't turn around and go back up, right? That defies gravity. Bacteria can't do that. And so they would settle in the neck. And so what he discovered was that if he filled these flasks with broth, which was something that can go bad, right? If I took a bunch of chicken broth and set it in my fridge, it's going to go bad, right? It'll get cloudy. It'll start to grow bacteria and mold. But if he boiled this broth and put it in these flasks, he left them for years, and they never grew bacteria. They remained sterile for months and months and months and months and years and years and years and years. And this was proof that the only way something was going to grow in there is if something else got in. That there was no life spontaneously generating inside this flask. The only way there was going to be any kind of growth or any kind of anything growing was if something was able to get in. And as long as he kept those flasks intact, nothing got in, because nothing can get past that swan neck. And the other proof to this was that the second he broke off the swan-necked flask, if he broke off the neck, bacteria and dust and viruses were able to sprinkle into the broth, and he would see bacterial growth within 24 hours, always. Okay, so this was proof, this was the definitive proof that life only came from other life. That life couldn't just spontaneously generate, that everything that existed on this planet came from something else. Or everything living that existed on this planet came from something else, another living organism. Okay? And that theory is biogenesis. Again, I wrote it up there, but the theory that life comes from other life. And this is a theory, like I said before, that is the prevailing theory until this day. And there is much evidence to prove it and none to disprove it in the 200 years since it was first developed. So as of now, this is our working theory, and it's the one that seems to fit the best with the experiments we have available right now. So yeah, and Louis Pasteur was a very famous scientist. He also developed some things that we'll talk more about later. He was the first one to develop pasteurization. That's where pasteurization comes from, which is a process where you heat up milk to kill bacteria. Arguably one of the most important health initiatives to ever happen. It arguably has saved billions of people's lives and prevented them from dying of food poisoning from drinking contaminated milk. So yeah, that was Louis Pasteur, right? And he was in 1859. So just kind of keep that date in your head. Again, I'm never going to ask you the date, but just keep that in mind. That was less than 200 years ago. That was not thousands of years in the past. This is not ancient history. It was less than 200 years ago. That's like 2 generations. Okay? All right, so another thing that was really important in the field of microbiology and the development of microbiology was being able to see the things that were growing. Okay? And that involves the discovery of the microscope. Now, the microscope was first developed in 1665 by a man named Robert Hooke. And he developed the first microscope strong enough to view eukaryotic cells. Now, remember from our very first slide, I think it was the first one, that eukaryotic cells are about 100 times bigger than bacterial cells. So his microscope, his very first microscope, was strong enough to view eukaryotic cells, which again are things like plants, animals, funguses, not bacteria. And he spent most of his time observing cells in plants and water. His most famous picture was a picture that he took through his microscope, right here. I'm gonna use a different color. That he looked through his microscope of cork on a tree. So he took a cork tree bark and looked at it through a microscope. And this was his most famous picture. It was put in Nature magazine, and it made him very, very famous. And he was actually the first one to coin the word cell. So as we refer to cells, because he saw individual little blocks in his cork, right? So those little squares, he called them cells. And that was what originally became, that's where the word cell originally came from. Now, Antony von Leeuwenhoek, about 20 years later, was the first to develop a microscope strong enough to view bacteria. So he was the first one to view bacteria and other smaller organisms than eukaryotic cells. He looked at his most famous things where he looked at drops of water, and he also scraped the plaque from people's teeth. So he was the first one to kind of make this connection that there were things growing inside of our bodies because he looked at the plaque on our teeth, which is covered in bacteria, if you didn't know that. Your mouth is absolutely teeming with bacteria. And plaque is just the bacteria forming little colonies all over your teeth. And so he looked at drops of water. He was the first one to kind of name bacteria, but what he called them was animalcules because they were molecules that moved or molecules that act like little animals. So he called them animalcules. He didn't know what they were. He didn't know what, the benefit of this was going to be or the impact of any of this was going to be. But he was the first one to see things moving in a drop of water that were way too small to be seen with the naked eye. Okay. Now, soon after microbes were discovered, when Louis Pasteur was kind of coming up with this abiogenesis theory, and we'll talk about Robert Koch and some other people that were kind of around the same time, most scientists and people came to this profound realization, and again, it's something that we take for granted now. We live in a world where we know that bacteria is everywhere, right? That's a thing we know. It's just the thing that happens, and it's been true for hundreds of years, so we don't question that. But can you imagine coming to the realization that you are covered in bacteria. There's bacteria inside your body, and there's bacteria. There are these little tiny living organisms living on every surface that you have ever touched in your entire life. And not knowing that before and learning that at some point in your life, it's a very profound, and I imagine probably pretty upsetting realization. But as soon as Louis Pasteur kind of came up with this abiogenesis theory, scientists everywhere realized that there were bacteria on every organism, on every surface, in the dust. There was no getting away from all of this bacteria. And that immediately translated into medicine, okay? Because we're talking about people being sick. So the first thing that people did was they immediately started looking at medical treatment and how that applied, how the bacteria and the viruses and the things that were living on all of these surfaces translated to taking care of humans, taking care of sick humans. So one of the first things that was developed was something called sterilization. And sterilization is a process by which we remove all living things from a surface. Right? And we all use that term sterilize, even if we don't really mean it. Like we say that like our dishwasher sterilizes stuff. It doesn't. We say that it does, but it doesn't really. Or even like if you're getting your ears pierced, they talk about sterilizing it. Or like if you're getting a shot, they talk about sterilizing the area. They're not actually sterilizing it. They're disinfecting it. But we'll get to what all that means later. But we use that term pretty normally in our everyday lives. This also includes removing something called endospores, which you don't have to write down right now. I just want you to keep that in mind when we talk about it later. In modern times, again, we think about sterilizing baby bottles. That's a really common one, right? People sterilize baby bottles, or you sterilize an area before you get an injection, or you sterilize a site before you have surgery, or things like that. But 200 years ago, these ideas were very, 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 very foreign. People didn't realize that there was stuff growing everywhere. And so this idea of having to clean everything was ridiculous. And the first person to really develop this set of sterilizing techniques, and that's what aseptic techniques are. Aseptic techniques are a set of techniques or a set of procedures designed to limit, we'll call it contamination. It's designed to limit the amount of bacteria or microorganisms that are moving from one surface to another. Okay? And we practice aseptic techniques a lot. You've probably experienced this if you've ever gone to the doctor, right? Doctors come in when they're going to stick their hands in your mouth or in any other part of your body. They put gloves on, right? Gloves are sterile. They wear masks. They clean everything, right? So aseptic techniques are just a set of procedures designed to limit contamination. Now, again, that's all very normal for us. We experience aseptic technique all the time. But the first person to really do this and to really take it seriously was a Hungarian physician named Ignaz Semmelweis. That's his name right there. And he was the first one in 1845 to really take aseptic technique seriously. And I think that his story about why he did this is really, really interesting. So I always tell this story. And Ignaz Semmelweis was a physician. He saw all kinds of patients, from people who were sick to, all kinds of different things. People at that time, a physician did everything, right? Physicians were dentists, and they took care of everything. But what he noticed was he was very observant, and he was able to notice that there was a difference in childbirth, specifically people who were giving birth, if they were attended by a physician while they were giving birth. So if they went to a doctor's office or a doctor saw them, a physician, they were, I think it was about, I don't remember the exact numbers, it doesn't really matter, but I think that they were almost 50% more likely to get an infection than somebody who was attended by a midwife, right? And if you don't know what a midwife is a specialized caregiver that is meant to deal with delivering babies, right? That is their specialty. They know all the ins and outs of delivering babies. And at the time, in 1845, a lot of times midwives were more highly utilized. Nowadays, most people go to doctors, whether that's good or bad, I'm not here to say that. But back then, midwives were much more common if you were delivering a baby. But he noticed that if a person giving birth was attended by a midwife, they almost never got an infection. But if somebody was attended by a physician when they were giving birth, they were almost 50%, almost twice as likely to get a postpartum infection and possibly die. It was like, why is that true? Why would that be true? Why would somebody who goes to a doctor be more likely to get an infection than somebody who's seen by a midwife? And he realized the answer is because physicians were hanging around with sick people before they went and delivered a baby, okay? It was very common for doctors, physicians, to perform an autopsy on somebody who had died and then go stick their hands in somebody else and deliver their baby. Okay? Now that's ridiculous, right? We don't do that. We wash our hands, we decontaminate, you're wearing PPE and all of these things. You don't transfer things like that. But at the time, hand washing wasn't a thing. Nobody knew what soap was, okay? So he realized that because midwives didn't see sick people, midwives only delivered babies. That was their whole job. And so because they were not performing autopsies or seeing sick people, they were much less likely to spread infections, right? It's a very simple observation. It's not even that hard to see. But if you don't know that bacteria is there, it's impossible to see that. How would you know that? And so he was the first one to make the connection that, oh man, these physicians are picking up these tiny little things that nobody can see from these sick people and giving it to the perfectly healthy person giving birth and getting their babies sick and getting the women sick. And so he was the very, very first physician that utilized hand washing. So he required anybody that worked in his hospital and himself that they washed their hands. They created a soap, lye soap, and they had to wash their hands and disinfect themselves between patients. And just the simple act of washing their hands in between all of their patients brought his mortality and infection rates down to the same level as the midwives. So just the introduction, this is way before gloves and all that stuff, but just the introduction of simply washing their hands brought the infection rates down to almost a negligible amount, which is pretty amazing and very fascinating. But 1845 was the first time that hand washing was introduced. And then that other person there, Joseph Lister, in 1860, he was the first surgeon to use aseptic techniques in emergency rooms or in operating rooms. So he actually developed a special spray. It was a disinfectant spray that he would spray all over every surface of his surgery suites. And so this was kind of the very beginning of sterile surgery. Right now, if you're having surgery, everybody in there is scrubbed up to high heaven. Everything is sterile. Nobody touches anything. You have to be scrubbed up. If you've ever had surgery, they make you wash yourself with special soap to get as much bacteria off your body as possible. They scrub you within an inch of your life. They shave all your hair off, right? Everything is as clean as humanly possible. But he was the first one to start doing that. And you can see it here. There's this guy right here who is spraying basically disinfectant over the surgery that's happening. Now, I don't know that we would do that now. That's basically Lysol that he's spraying into somebody's open body cavity, which doesn't seem like a great idea now, but it did cut down on the amount of post-surgical infections. Because back before hand washing and surgeries and things like that, anybody who had to have any kind of procedure done was more likely to die than not, because they got an infection almost every time. Because nobody was washing their hands, nobody was using any kind of disinfectants, everything was just a free-for-all. So he was kind of the beginning of the idea of surgery suites and cleaning everything as much as possible, use strong chemicals. And his name is actually, Joseph Lister is what the original Listerine is named after. So Listerine is mouthwash, and the first Listerine was the same chemical that Joseph Lister used to disinfect his surgery suites. Now Listerine is not that chemical anymore. Again, it's basically Lysol. It's called a phenol. It's basically Lysol. Swishing with Lysol is probably not a great plan, but that's what Listerine is named after. Right? So the germ theory of disease, all of this kind of work of Louis Pasteur and Robert Koch, who we're going to talk about in a minute, and the works of Semmelweis and Lister, all kind of coalesced together to come up with this germ theory of disease. And the germ theory of disease is simply that infectious diseases, is that infectious diseases are caused by microorganisms. So these organisms that are too small for us to see. For a long time, people thought that infections and disease were caused by gods or by karma, or doing bad things in your life meant that you were going to get infections, or breathing in bad air, or being cursed by a witch, right? There's all different kinds of theories about where diseases used to come from. And in 1860, 1870, 1880-ish was when this theory was actually developed, that diseases are not caused by being cursed or because you made a god mad or anything like that. It was because you caught a bacteria. Okay? And Louis Pasteur was the first scientist, again, that definitively proved that microorganism could cause human disease. He would isolate specific bacteria and introduce it to a healthy host, like a healthy mouse and things like that, and observed that when you took this bacteria and you put it in this mouse gets sick. Okay? And he developed pasteurization, like I said, a process that completely revolutionized food safety. It was the first time that anyone was really doing anything to preserve food and try to make food more safe. And like I said before, he arguably prevented the deaths of billions of people by inventing pasteurization. And for those of you who don't know, pasteurization is just heating up milk to slightly below boiling. So that's not even all that revolutionary, right? But you just have to put yourself back in that time when this was revolutionary, this idea that there was bacteria everywhere, and that drinking raw milk was drinking cultures of millions of bacteria that could potentially make you really, really ill. And then Robert Koch developed something called Koch's postulates. Which we're not going to talk too much about, but Koch's postulates is a series of ideas and steps that talk about if a microbe is able to cause disease, so we have this bacteria, is it capable of causing disease, and what disease does it cause? And it's a series of steps that you have to go through to prove that this bacteria causes this disease and how we know that. So he was the first one to identify a lot of diseases. He was the first person to identify what caused tuberculosis, which is a huge infectious disease, anthrax, smallpox, I believe, all kinds of different organisms. Right? So that's medical microbiology. Now we're going to kind of change gears here a little bit. So we went through what a microbe is. We went through how they were discovered, how the first people knew that they existed. Now, we're going to talk a little bit about the things that make up living cells. So I've used that word cells a bunch of times. Now we're going to talk about the things that exist in a cell. Now, we are going to cover, just something I want you to be aware of as we do all this, is we are going to cover a very small portion of what makes up a cell, right? What makes up a cell could be multiple classes. In fact, there are multiple classes. They're called cell biology, right? Where you talk about all of the things that happen inside of a cell. It's fantastically complicated. And we're not going to go into all of the details. So all you have to know on these subjects is what I talk about in class, okay? And I've tried to set your SmartBook assignments to only cover the things that we talk about, but if something slips in that I missed, or you're reading your textbook, like if you're just reading your book, like she didn't talk about this at all. I know. If I don't talk about it, you don't have to know it, okay? Reading the book is not a bad idea. But I did try to pare it down a little bit and try to keep it as simple as possible, and try to keep it to the information that you have to know. So if I don't talk about it, you don't have to know. All right? So the first thing we're going to talk about as it comes to cell biology and what makes up cells are things called macromolecules. Now, macromolecules, again, if you look at the word macro, what does that word macro mean? Large, big, right? So macromolecules are large molecules. There's big molecules. That's it. Okay? And there are 4 major macromolecules that exist in all cell types. Now, most people find this easiest to think about in terms of humans, because when you think about how humans exist, we require all 4 of these macromolecules too. Okay, so there are 4 macromolecules that are necessary for the survival of any type of cell. And they are, over here, they're carbohydrates, which is a fancy name for sugar. Carbohydrates is the first one. Lipids are the second one. This is a fancy name for, does anybody know? Fats. Yep, that's a fancy name for fats. Nucleic acids. Does anybody know what that one is? That one's a tough one. That is DNA. So we'll talk about what all of these do later. DNA is the other one. And then protein, which doesn't really have a fancy name. But 3 of those are things that are required in our human diet, right? To survive as a person, you have to have proteins, fats, and carbs to live. That is what your body requires to metabolize, to do things. And the same thing is true for all cells. From bacteria all the way up to the largest animals, all cells that exist require those 4 macromolecules. Now, macro means large, just like we talked about. And all of these large molecules, another word that we use for them is a polymer. And poly as a prefix means many, right? Poly means many. So many big molecules. It's many things all hooked together, right? So these big molecules, these big macromolecules are polymers, macromolecules. And every single one of those macromolecules is made up of a smaller component, okay? And that small component, or the individual component, and we're going to talk about all of these individually, so don't freak out yet, but the individual component is called a monomer. Mono meaning one, right? Mono means one. Like monogamous, right? Monogamous means you have one partner. Polyamorous means you got many, right? Mono means one, poly means many. So a polymer or a macromolecule is a combination of many monomers. I kind of like to think of them as like Lego towers, right? A polymer or a macromolecule is a Lego tower. Well, how is a Lego tower built? You take one Lego and you stack it on top of the other. And you stack it on top of the other, stack it on top of the other. The Lego, the individual Lego brick, is the monomer. The tower is the polymer. And each one is made up of a different type of Lego. Okay, so carbohydrates, and we're going to talk about these individually. But I have the monomer paired up with the polymer here. So the monosaccharide, when you stack them, all the monosaccharides together like Lego bricks, you make a carbohydrate. Okay, we're going to go through each one of these individually though. But there is the overview. All right, so we're going to talk about each one of these individually. The first one we're going to start with is carbohydrates. Again, carbohydrates are also known as sugars. They are also known as saccharides. Those are just all of the different names that we give them. Okay, carbohydrates, also known as sugars, also called saccharides. Okay, and like I said before, every single polymer, so the carbohydrate is the macromolecule. That's the polymer. That's the Lego tower. The monomer is the individual bricks. So for carbohydrates, the monomer, the individual brick, is called a monosaccharide, which again, mono means one, saccharide means sugar. Monomer, monosaccharide, one sugar. Okay. Now, monosaccharides by themselves can be used as energy or food. So examples of monosaccharides are glucose, right? That's our main, as humans, glucose is our main food source. That is what's in most of our food is glucose. Lactose is another one. Lactose is milk sugar. And then sucrose. Sucrose is another one. It's found in a lot of fruit and other types of food. But all of those monosaccharides individually by themselves are used as food. That is what our body consumes and breaks down and uses for energy. Okay, that's why when you eat Snickers bars, you get a sugar high, right? We call it a sugar high. But it's really just that you have lots and lots and lots of energy because candy and foods like that have tons and tons and tons of sugar. They have a lot of glucose. All right, now when you take any of those monomers, so you can take glucose or lactose or sucrose, the most common one is glucose, but you can take them, that one sugar molecule, the monosaccharide, and link it together with a bunch of other monosaccharides, right? We're like building a Lego tower. We're taking hundreds and hundreds and hundreds, if not thousands and thousands and thousands of those individual monomers, those individual monosaccharides, and hooking them together to make a big polysaccharide. Okay, so we're making a big Lego tower out of the individual Lego bricks. Now the polymer, again, is called a polysaccharide. Polysaccharide, many sugars, many. And again, this is also called a carbohydrate. So it's confusing. I don't know why we have to give everything so many names. We just do. Okay. Now, the polymers are actually more used for structural components. We'll talk about that in a second, and energy storage. So rather than being what our body uses for energy, the polysaccharides is how humans and other types of organisms store their energy and save it for later. So what happens, a lot of people find it easier to talk about what happens inside of our bodies because we can kind of relate to that more. So what happens inside of our body, say you eat your breakfast, right? Now, our body is constantly burning energy, right? We're talking, we're breathing. Even if you're not moving, you're existing, right? Your body is breathing, you're moving muscles, your heart is beating, your brain is sending signals to the rest of your body. Your body is doing stuff. You are using energy all of the time. Now, do we as humans eat all of the time? Do you have to eat 100% of the day? No. And the reason for that is when you eat your breakfast in the morning, your body takes all of that sugar that you have eaten, all of the glucose mostly, we'll just say it's all glucose, takes all that glucose and it uses what it needs right then. It's like, okay, I need to do this, I just woke up, I haven't eaten in a while, and it'll do the things that it needs to do. It uses the sugar that it needs. It takes a little bit of it and uses it right now. And then the rest of it, it says, I don't need this right now, but I'm gonna need it later. So what it does is it takes all those glucose molecules, the individual monomers, hooks them all together, and makes this big structure and sticks it in your liver and saves it for later. Okay, so it's taking that energy and putting it in a storage closet to wait until later when we need it. Okay, it's like when you come home from Sam's Club with your giant thing of paper towels. You don't need all those paper towels right now. You take the one that you need and you put the giant package in the garage and you save it for later. Okay, that is what your body does. You take the glucose you need for now and then you hook all of them together into these big polymers and stick it in your liver and save it for later. Okay, so that is where your energy is stored. And then as you go throughout your day, your body will take a little bit out of the liver and break it back down into monomers and use the energy that way. All right, now that storage form that we put in our liver is called glycogen. So in humans, humans and other animals, the storage form that we use is called glycogen, and that is what is in our liver. We store a bunch of sugar in our liver to use as we go about our days. Okay, that is the storage form. It is a polymer, a polysaccharide. Okay, starch, and you can probably tell, like, where do you get starch from? Can you think of something that's starchy? What has a lot of starch in it? Potatoes is a good one. Corn is also really starchy. So starch is a very similar thing to glycogen. It's a storage form of glucose, but it's mostly utilized by plants. So this is storage in plants. And that's what potatoes are. Potatoes are actually just the place that that plant has put all of its sugar to save for later. And then we very rudely pick it and take it and eat it because they're delicious. But that is where plants store all of their extra sugar, because plants know that they are not going to have, right, they're going to reach a winter time, there's not going to be food storage available. That is where potato plants shove all their extra sugar. They make these polymers called starch, and they stick it in a vegetable. And it's the same thing with a lot of other vegetables, a lot of other plant products. Like celery, corn is another good example. Legumes, all kinds of beans, they have lots and lots of starch in them as well. Okay, now chitin and cellulose are actually structural components, because what can happen is when you make these giant structures of sugars, even something as small as like a grain of sugar. Like a grain of sugar is a physical thing, right? That's a bunch of glucose molecules all hooked together to make a single molecule, a grain of sugar. And grains of sugar are hard, right? And if anybody's ever seen, like when they melt sugar, you can make all kinds of crazy stuff with sugar. Sugar, when it's in the correct form, can be very structural, can be hard. So what can happen is these polysaccharides, some of them are used as structural support. One of the most common ones is something called chitin. Chitin is a sugar that is found in the exoskeletons of beetles. Okay, so what the beetles do is they take all of these individual sugars, they hook them together to make a polymer, and then they basically use that as armor on the outside of their body. The entire exoskeleton of beetles is made out of sugar, which is pretty crazy if you think about it. But chitin is that substance that you find. That's what makes beetles crunch when you step on them. Okay, cockroaches too, even spiders. But chitin is a very, very hard substance. Another one that provides structural support is cellulose. Now, cellulose is the structural support in plants. If you've ever had, you know in celery, you know that the little strings that you can pull apart in celery, that's all cellulose. And the reason that it exists, it's hard, and it provides that plant with structure. That's part of why plants can stand straight up, right? Is because they have this sugar that's stacked on top of each other and makes a stiff and structurally sound support system. Parts of stems and leaves and bark, tree bark and stuff like that is all made out of cellulose. So all of that is just sugar. Okay, so sugars can do a lot of different things. The monomers, again, the big important thing to remember there is that the monomers are used for energy and food. That is what gives organisms energy right now. The polymers are mostly energy storage and sometimes structural components. Okay. All right, next we have lipids, which, as you guys said, is a fancier name for fats. And we have a couple of different types of fats. We're going to talk about each one individually. The first big group of fat molecules are called triglycerides. And the biggest purpose of triglycerides is what we call long-term energy storage. Now again, we're going to talk about what happens inside of a human. So say you eat your breakfast. Your body uses the glucose that it needs now, right? It uses the monomers. We just talked about that. It makes a bunch of polymers called glycogen, and it takes that glycogen and puts it in your liver and saves it throughout the day. Well, the problem is that your liver will only hold so much glycogen. So if you are consistently eating more sugar than your body needs, eventually your liver will run out of room. And your body's gonna go, what do I do with all this extra stuff? I got too much sugar. I don't have anywhere else to put it. The glycogen's out. We don't have no more space in the liver. What do we do with it now? And so what your body will do then is it will create triglycerides, which in humans forms adipose tissue, which is fat tissue. Okay, so what your body does is if you are consistently eating more sugar than you need, as soon as your liver is filled up with glycogen, your body will take all of those extra triglycerides or all those extra sugars, and it will turn them into fats. It'll take those sugars and turn them into triglycerides and turn them into fats. And those triglycerides will become adipose tissue. And then your body will basically shove it wherever it can, right? Shoves it on all parts of your body. And so this is what we call long-term energy storage. The glycogen is what we call kind of short-term energy storage. The glycogen in your liver is usually put there and used within a couple of days, right? Usually within the day if you're eating properly. It will be used within the day. But if you, and so you'll save all of those extra glucose, all of that extra sugar and energy in the form of adipose tissue, which will stay there for a long period of time. And we call it long-term storage because your body only uses it as a last resort. So your body will actually only break down adipose tissue if it goes through all of the food you've currently eaten, and then it will eat through all of the stuff in your liver. And then it's like, oh no, we're out. Now we got to go into deep storage, right? And then it'll start breaking down that adipose tissue and using that for energy, which is a state, if you're using your adipose tissue for energy, it's a state called ketosis. That's a word that is kind of buzzy right now, being on a keto diet and being in ketosis. And what that means is you're using energy from your fat stores rather than your glycogen. So triglycerides are long-term energy storage. And triglycerides, the way that they are made up, they're called triglycerides, and they are made up of a single glycerol molecule. Now, just to clear this up, I don't think I've said it yet. You're not going to have to know how to draw these, okay? I'm never going to ask you to draw a carbohydrate or draw a lipid or draw a triglyceride. You don't even have to recognize it, right? I'm not even going to show you a picture of a glycerol and say, tell me what this is, okay? But you do have to know the components. You have to know that carbohydrates are made out of monomers, and you have to know that a triglyceride is made up of a glycerol and 3 fatty acids. But you don't have to recognize those things or draw them. Does that make sense? What I'm expecting of you? Okay, so this is made up of a triglyceride molecule is made up of a glycerol and 3 fatty acids. So this is, I don't know if it works well, it does work, okay. So this is a glycerol right here, okay? And notice it's got 3 carbons, 1, 2, 3, with 3 little OH groups that hang off the side. Okay, so 1, 2, 3. So a glycerol molecule has got 3 nice spots for something called a fatty acid to attach. Now this fatty acid is made from excess sugars. So sugars are all made up of carbons and hydrogens. So when your body has excess sugars, it does a lot of things that we don't have to understand and turns those excess sugars into fatty acids. Okay, and then each of those fatty acids, notice that these fatty acids also have an OH group on the end, and those 2 OH groups are going to combine so that they become attached. Okay, so 1, 2, 3. We have 3 attachments, and what gets left behind is this H here, and this O there, which is H2O, which is water. So what will be removed is water, and what you end up with is this triglyceride molecule. This is the glycerol up here, or what used to be the glycerol, and now it has these 3 fatty acid or hydrocarbon chains attached to each one of those groups. Okay. Fatty acids and hydrocarbons are the same thing. We use those names interchangeably. All right. Now, we do need to talk about the difference between, this is a little confusing, but we got to talk about it. The difference between saturated, and these are words that you've probably heard before, but the difference between a saturated fatty acid and an unsaturated fatty acid. So saturated fat, unsaturated fat. Just in terms of food, right? We talk about saturated fat being bad, and you want to limit the amount of saturated fat you have, versus unsaturated is like better. And we need to talk about why they're different and why one is considered better than the other. So what saturated fatty acids are, so this is a saturated fatty acid on the left here. And we call it saturated because notice all of these lines in between those carbon molecules, right? The C's are the carbons. Notice that all of those lines in between the carbons, there's one line. Okay, that's called a single bond. And we call it saturated because each and every one of these carbons has 2 hydrogens attached. Okay, so notice that each one of these carbons, if I look, they've all got 2 hydrogens attached. Okay? On this side, what that means is on the saturated side, the carbons are saturated with hydrogens. They are full. Each of them has 2. They cannot have any more hydrogens. There are 2 hydrogens, that's all they can handle. Okay, but over here on the unsaturated side, we have added, notice that some of these carbons have 2 lines between them. And what that means is you are taking up one of the spaces that carbon has by binding it to carbon a second time. Now I know that's a lot of things, but we don't have to understand the details. What that means is that, notice these 2 carbons here, each only have 1 hydrogen. So they are not saturated, they are unsaturated. They don't have as many hydrogens as they could have. They could have 2, but they only have 1. Okay, and the same thing is true down here. When you add a double bond like that, you take away one of the hydrogens from each of the carbons. So those are unsaturated fatty acids. Now what that does, if you look at this picture, and I know it's weird to think about this stuff in reality, but this is what is happening on a microscopic level, is saturated fatty acids form straight lines. Okay, those single lines, like you can see here, they form straight stick-like lines. Okay, unsaturated fatty acids, along those double bonds, they physically bend, like you see here. They will have kinks in them. Now, think about, we have tons and tons and tons of these triglycerides, and I tell you that you have to fit as many triglycerides as possible into a single space. Okay, now think about it like our bodies. If I was a triglyceride and I was standing like this, if I had to fit as many people as I could on this platform right here, am I going to be able to fit more things if I'm standing like this or if I'm standing like this, right? If there's a big old kink in it, there's not going to be as much room. If I tell somebody to come stand up as close to me as possible, and I'm standing here like this, somebody is going to be able to come up and stand right next to me, right? They'll be able to shoulder to shoulder, stand right next to me. If I've got my legs sticking out, they're not going to be able to get as close. So in saturated fatty acids, those triglycerides line up in straight little lines. They line up really close. And what that results in is a more solid, substantial substance. So saturated fatty acids are the ones that tend to be more solid. So a saturated fatty acid would be like butter. Okay? Because at room temperature, butter is a solid. And that's because all of those little fatty acid chains are lined up in nice little straight lines because they're all saturated. And so they're all following the rules and standing there in straight little lines, and they form a nice, solid, substantial structure that is solid at room temperature. Unsaturated fatty acids are things like olive oil. Because olive oil at room temperature, is it a liquid or a solid? It's a liquid. Avocado oil is a liquid at room temperature. Coconut oil, sometimes. Coconut oil is a little more touchy. But the ones that are liquid at room temperature, they're liquid at room temperature because they're standing next to each other like this. And there's no room for anybody. And so everything is a lot more fluid. They are not stacked on top of each other like military ranks, right? They are more free-flowing. They're fluid, okay? So that is the difference between a saturated fatty acid and an unsaturated fatty acid. Okay, saturated fatty acids are the ones that are supposed to be worse for you because they are more solid. They're supposed to clog up your arteries because they form those solids inside your body. Unsaturated fatty acids are more liquid because of the unsaturated nature, because the double bonds create kinks. Right? I know that's a lot. It feels like we just jumped into this, right? Now, one other thing that we need to bring up about fats in general is that fats are what we call, and this is true for almost all fats, including triglycerides, but fats are what we call hydrophobic. Okay, now again, look at the word. What does phobic mean? Fear or afraid of, being afraid of something. What does hydro mean? Water. So hydrophobic means water-fearing, afraid of water. What that means is that fats in general do not mix well with water, right? And anybody who's ever tried to wash peanut butter off of a spoon is going to know that, right? Because peanut butter is all fat. And when you try to run water over it, it doesn't do jack because the water cannot get into the peanut butter. You have to use soap, right? It's not going to rinse off. Or if you've ever tried to make a salad dressing, right? Even if you take a bunch of olive oil and you pour a bunch of water in it, even if you shake it up and stir it up and whatever, you let it sit again, the water and the oil is going to separate right back out, okay? Because oil and water don't mix. Because oils and fats are hydrophobic. Hydrophobic. The other side of that is something that is hydrophilic. Philic means loving. I think I have that written down somewhere else. But hydrophilic is the other side of that. Now, our next category of lipids are called, and remember these are all fats, so our next category of lipids are called phospholipids. They are called phospholipids because they have a phosphate, right? This is going to be a recurring theme, is that scientists are not super creative. So if you ever don't know what something does, look at the word. The word is going to tell you what it is. This is called a phospholipid because it's a lipid that has a phosphate in it. That's not that complicated, okay? But what that means is that instead of having, so over here we had a glycerol with 3 fatty acid chains. This entire molecule is a fat, and it is all hydrophobic. Everything about this molecule, the triglyceride, is hydrophobic. With a phospholipid, the difference here is that we actually still have a glycerol molecule, but in the previous one, we had 3 fatty acid chains being attached to the glycerol, right? The glycerol has 3 spots for things to bind. Instead of 3 fatty acid chains, we're going to get 2 fatty acid chains. So there are 2 fatty acids. And that leaves 1 more spot for something to bind, right? Glycerol has 3 spots. So 2 of those spots are going to be fatty acid chains, and 1 of those spots is going to be a phosphate. Now, what this means is that the bottom part here, these fatty acids, are going to be, again, everything has to have multiple names. Nonpolar also means hydrophobic. Okay, so those fatty acids, just like we talked about, fatty acids or fats are hydrophobic. So the bottom part, where the fatty acids are going to be hydrophobic. But this phosphate group is actually hydrophilic, or charged, or polar, okay? So this is, did I say hydrophobic again? Hydrophilic. Okay, so the top of this molecule is hydrophilic, which means that the top does like to interact with water. Okay, so we have a single molecule here that the top of it is hydrophilic, meaning it will combine with water, and the bottom is hydrophobic, meaning that it won't. We have 2 separate pieces here. And this phospholipid is specially designed to form membranes, which are the outer surface of all cells. So all cells have a membrane, sometimes called, we'll call it lots of names, a plasma membrane, a cytoplasmic membrane, all kinds of stuff. But the membrane is, we'll just call it a membrane for now, is the outer boundary of every single cell that exists in the world. And the reason that these work and what they do is because of these very, very special molecules that have a hydrophilic head or a hydrophilic top and a hydrophobic tail, because they're made up of a glycerol, 2 fatty acid chains, and a phosphate group. Because just like I said, if you took a bunch of water and a bunch of oil and you poured them together and you shook them up as hard as you could and you let it settle, the oil is going to separate from the water. And the reason it does that is because the oil doesn't want to touch the water. So it arranges itself so that as little of it is touching the water as possible. Okay, it's going to rearrange itself until as little of it is touching because it doesn't like it. It's afraid of it. So what will happen with these molecules is many times these hydrophobic tails and hydrophilic heads arrange themselves so that the hydrophilic part is touching the water and the hydrophobic part is not. The phobic part gets to hide while the hydrophilic part touches the water. So this is a membrane right here. And on either side of this membrane will be water. One of these will be inside the cell. One of these will be outside the cell. And this phospholipid membrane is what's separating the inside of the cell from the outside of the cell. It is the barrier between the 2 places. And you'll notice that all of the hydrophilic heads, which are these yellow circle parts, have lined up so that they are touching the water, and then the tails stick out into the middle where they're not touching any water at all. Okay? And that is what separates the outside of a cell from the inside of a cell, are phospholipids. They often form these things, like it says right there. I don't have an eraser. Called a phospholipid bilayer, right? Bi meaning 2. Okay? Because we have 2, in the phospholipid bilayer, we have 2 layers, right? We have 2 lines of hydrophilic heads with the tails in between. And this hydrophilic, this phospholipid bilayer is what every cell in the world that exists on this planet is made out of. Okay? All cells have some type of membrane that looks just like this. Okay? And we'll talk more about this later, but a lot of these membranes have things in them that allow certain things in a cell, keep certain things out, but that's a more complicated subject that we'll get to later. Okay? All right, so our next type of lipid, we have a couple. The first one are called sterols. So sterols are pieces of fats. Again, these are all lipids, and they are used to provide cell membranes with strength, and they are also used in animal hormones. So these cholesterols, or sterols, they are stuck in, you can kind of see them here, they are stuck in this plasma membrane to kind of give it some structural support so that it's able to be the way that it is. And we'll get more into that later, but that's all you have to know about it for now, is that sterols are used to provide structural support and strength to cell membranes. The most common sterol in animals is called cholesterol. Right there, I was gonna write it, but I already wrote it. It's called cholesterol. There are other types of sterols that exist in bacteria and things like that, but we're not gonna talk about their names. Animals have cholesterol, right? That's what that's what you have to get tested. I know all of you are nice and young and all of your organs work the way that they're supposed to right now. Once you hit about 30 or 40, you're gonna have to start getting your cholesterol checked because things stop working. It's a real fun time. But you don't wanna have too much cholesterol, but you have to have just the right amount, okay? Because it provides strength for your cell membranes. Cholesterol is necessary. I know that cholesterol gets a bad rap, like right here, you don't want your cholesterol to be too high, don't eat too many egg yolks because you're gonna get too much cholesterol, but you need some, you just don't want too much. So it can kind of be a balancing act, okay? And then the other thing that sterols do, including cholesterol and other types of sterols, is they form the basis of our animal hormones. So in humans, does anybody know what our two major steroid hormones are? They kind of have the word steroid in them, or roid. Testosterone is one. And estrogen. Okay, so our steroid hormones are animal hormones, and there's other ones that are also in this category, but testosterone and estrogen are the big ones, which are our sex hormones, right? They're what allow humans and other animals to reproduce. If we didn't have testosterone and estrogen, our species would cease to exist because we wouldn't be able to reproduce. So sterols are not all bad. Like I said, they get a bad rap because a lot of people eat too many, but they are a necessary thing. They are a thing that we all have to have. But those are their two major purposes. They provide strength in membranes, and they serve as the basis for steroid hormones. And then the last one is our waxes. So waxes are another type of lipids that provide water repellent. And there are a lot of different types of waxes. In fact, you have a bunch of wax on your body, right? I know it's weird to think about, but your skin is water repellent, right? You don't absorb water. Your skin has wax on the outside to keep water out. There are a lot of things that have waxes, but there are a lot of bacteria that have wax on the outside of their bodies, specifically something called mycolic acid. This is found in the membrane of certain bacteria. Specifically, a bacteria, the most common one, is the bacteria that causes tuberculosis, or TB. And we're going to talk about tuberculosis a lot as we go through this class, mostly because I know it may not seem like it because of the country that we live in, but worldwide, tuberculosis causes more deaths from infectious disease than anything else. And the reason you may not realize that is because we live in a country where it's not that common. But in other countries, it is very common, and there are lots of countries that can't afford to treat people for it. And so there are still millions and millions and millions of people that die of tuberculosis every year. And And so we'll kind of talk about it a lot. But one of the reasons that tuberculosis is so dangerous is because their membranes have this waxy layer on the outside, which actually makes it really difficult for the human immune system to kill. So the human immune system has a really hard time dealing with the waxy layer, that mycolic acid wax on the outside of tuberculosis. All right, I think that we'll call it there, and we'll start up with proteins next time.